Many dental procedures are potentially painful. If the nerve impulse reaches the patient's brain, it's interpreted as being painful. How does the nerve conduct this impulse to the brain, and how do local anesthetics act to prevent this from happening? Sensory neurons that conduct pain are composed of three major parts: the axon, the dendrite, and the cell body. The single nerve fiber, or the axon, is a long cylinder of axoplasm which is encased in a thin sheath, the nerve membrane, which separates the axoplasm from the extracellular fluids. In some nerves, this membrane itself is covered by an insulating lipid-rich layer of myelin. There are constrictions along the myelin called nodes of Ranvier. At these nodes, the nerve membrane is exposed directly to the extracellular fluid. At rest, there are differences in the concentrations of ions found inside and outside the nerve. The inside of the neuron is relatively negatively charged compared to the outside of the nerve. When stimulated, sodium channels open, permitting the rapid influx of sodium ions into the interior of the nerve cell. The interior of the cell becomes positively charged as these sodium ions rapidly enter into the cell, a process we call depolarization. Then the process of repolarization starts, with the active transport of potassium ions out of the nerve cell to restore the resting equilibrium. In non-myelinated nerves, the impulse moves forward by a sequential depolarization of short adjoining membrane segments. In myelinated nerves, the impulse leaps forward from node of Ranvier to node of Ranvier, a much more rapid conducting process called saltatory conduction. The propagated nerve impulse reaches the brain, where it is interpreted by the patient as pain. How do local anesthetics act to prevent the nerve impulse from reaching the brain? Locals act by blocking sodium channels, so that when the nerve is stimulated, sodium ions cannot enter into the cell, thereby preventing depolarization. The local anesthetic is deposited as close as possible to the nerve and by diffusion enters into the nerve. Local anesthetic outside the nerve diffuses into an uncharged and a charged ionic form. It is only the uncharged form that is lipid soluble and is able to diffuse through the nerve membrane and into the nerve cell. Outside the nerve, only charged ions remain, and some redissociate into uncharged ions which can then enter into the nerve. Theoretically, all the charged ions eventually become uncharged, but because there are lymphatics and blood vessels in the surrounding tissues, a quantity of local anesthetic will never enter into the nerve. On the inner surface of the nerve, some of the uncharged ions redissociate into the charged form. These charged ions enter into sodium channels attaching to drug receptor sites, blocking the entry of sodium into the nerve when it is stimulated. This prevents the propagation of the nerve impulse. The impulse does not reach the brain, and pain does not occur. When enough local anesthetic is diffused into the nerve, impulse conduction is stopped and the patient is anesthetized. How do local anesthetics stop working? Eventually, an equilibrium occurs between the amount of anesthetic inside and outside the nerve. As the anesthetic outside the nerve continues to diffuse away from the nerve in blood vessels and lymphatics, the concentration gradient changes, with less anesthetic outside than inside, and the local anesthetic begins to diffuse out of the nerve. Eventually, there is not enough local anesthetic left within the nerve to adequately block nerve conduction, and the patient slowly begins to feel again. This process is termed redistribution. The local anesthetic is still in the patient's body as an active drug, but is simply not in the nerve in a high enough concentration to fully block nerve conduction.